Assalamualaikum and salam sejahtera. I am ALIDA MAHYUDDIN from the Faculty of Dentistry UKM, and the title of my study is Compound Identification and Biocompatibility of Effective Eco-Produce on Mouse Fibroblast. Enterococcus faecalis is commonly found in persistent root canal infections. It is a normal inhibitor of the oral cavity. However, it is able to survive the harsh condition in the root canal system. During root canal treatment, several methods are used to remove the biofilm structure, which includes canal shaping and effective irrigation of the root canal. The gold standard irrigant has always been sodium hypochlorite, but it has several disadvantages as listed. There have been many attempts to come up with an alternative irrigant that is able to overcome the shortcomings of sodium hypochlorite. Most of them are plant-based materials. One possible option is the effective eco-produce. This is an organic product from various kitchen waste, molasses and water in a ratio of 3:1:10, fermented for more than 3 months. It is created by Dr. Rasukon, who is a Thai farmer. Its common name is garbage enzyme, although it actually consists of enzymes, organic acids, and other trace elements. Arun and Sivan Shanmugam in 2015 found that various fruit and vegetables AEP contain protease, lipase, and amylase. Various sources of EEP also vary in terms of hydrolytic enzyme activities. They concluded that EEP of pineapple peels, orange peels in 6:4 ratio possess the highest degree of hydrolytic enzyme activity. EEP is also known to contain various organic acids. Some studies also found EEP to have antibacterial properties against pathogens such as E. coli and Staph aureus. On the left side of the slide are the studies that have been carried out by our research team from Faculty of Dentistry UKM. As a summary, we agreed with Arun and Siva Shanmugam that EEP from mixed source materials, pineapple and orange, showed the most promising potential as alternative endodontic irrigant. Therefore, the aims of the present study were to determine the constituents of EEP of 3 and 6 month mixed pineapple and orange peel and the cytotoxicity of EEP against mouse fibroblast. For the materials and methods, the EEP was prepared according to Prakash 2011 protocol and fermented for 3 and 6 months. The solutions were then sterile filtered and prepared into various concentrations. The EEP was then separated by LC-MS. Multivariate analysis with principal component analysis or PCA was performed. As for the cytotoxic testing, L929 cells, which are mouse fibroblasts, were selected. Fibroblast is the main type of cell in the dental pulp tissues. MTT assay was used. The colorimetric absorbance value reflects the number of surviving cells and metabolic activity. IC50 value is a half maximal inhibitory concentration that measures the potency of the sample in inhibiting 50% of cell viability. For the results, Figure 1 shows the PCA score plot of 3 months and 6 months EP separated into a cluster distinct from each other. Figure 2 shows the loading plot of chemical compounds, which represent the contributing weights of each compound to PCA1 and PCA2 respectively. Figure 2b shows the distinctive chemical compounds in the samples in 3 months EEP. In Figure 2c, the distinctive chemical compounds of 3 months and 6 months EEP are shown. And lastly, Figure 2d shows the distinctive chemical compounds in the samples of 6 months EEP. Other possible compounds of the same molecular weight that may impose an antibacterial effect on AEP are listed in this table. Stachydrain is the main compound found in both specimens that has been shown to inhibit the growth of E. coli and Staph aureus at the MIC of 5 mg per ml. In addition, D-ioditol, also found in both specimens, is a type of sugar alcohol which has been reported to have a bactericidal effect, particularly on Streptococcus species. Compared to the study by Arun and Siva Shanmugam, Our study did not show a large number of organic acids, such as acetic acid or lactic acid. Our findings were mostly on sugar alcohol instead. Table 1 and 2 show the solubility and the IC50 value for both specimens. As the MTT assay only measures the mitochondrial activity of the cells, in order to determine its viability, the results might not be totally representable as the actual number of viable cells. This is because some cells can still survive without the mitochondria to undergo the anaerobic respiration. In this study, therefore, although the IC50 of 3 months EEP is higher than the MIC of 50% concentration, suggested to exert an antibacterial effect as found by Ng et al. In 2019, the MTT assay might not be 100% accurate to determine that 50% EEP is cytotoxic to mouse fibroblast tissues. Furthermore, root canal reagents are aimed to be used within the confines of the root canal system. Sodium hypochlorite is also found to be cytotoxic to cells of dental origin, even at a very low concentration of 0.05%. We recommend that further studies are to be conducted to validate the results of the cytotoxic testing. We conclude that 3 months and 6 months mixed pineapple and orange 6:4 ratio EEP are composed of many chemical compounds. Its antibacterial and antibiofilm activities could be due to iodotor, stachytrin, choline, and pentolactone. We cannot conclude that EEP is not safe to be used as an endodontic irrigant. Here are my references, and with this, I thank you.